Going to cover today skeletal muscle glucose uptake during exercise. We'll also talk about it at rest and really highlight how there are some differences when it comes to the signaling between the two and why we'd expect them to be different. So molecular mechanisms by which exercise directly increases glucose uptake is largely a little bit elusive, but we've got a lot of the current topics regarding regulation of glucose uptake have derived from efforts in just insulin signaling field itself. So we're going to start with just talking about insulin signaling, how it elicits changes in glucose uptake. I've labeled that here the insulin signaling cascade. And this originated because we started with this insulin signaling cascades. A lot of efforts, if you look at the literature over the last 20, 30 years, a lot of efforts been to determine if any of the components of insulin signaling cascade are also what mediate exercise-induced responses of glucose uptake. So if you're dealing with anybody who's concerned about glucose uptake and regulation of blood sugar, which if you're going into medicine is going to be a large percentage of your population, these things are going to come into play. So let's just start with the insulin receptor itself. And what are some characteristics about it that we need to know? Well, it's a heterotrimetric membrane glycoprotein. It's got 2 alpha and 2 beta subunits. So we can see here in this image, we've got our plasma membrane. We've got 2 alpha and 2 beta subunits. With insulin, it binds to the extracellular alpha subunits, leads to activation of the transmembrane beta subunits, and we get autophosphorylation of the receptor. So hopefully you can apply this to what we learned in the last module about the different types of receptors. We're talking about, it's very similar to our tyrosine kinase, where it can self-autophosphorylate. So we get our insulin molecules bind to our receptor. We've got this autophosphorylation occurring here, so we get this phosphorylation of our membrane or of our receptor. And the predominant mechanism by which this insulin receptor then sends this signal onward is with the recruitment of the insulin receptor substrate docking protein, IRS protein. So we've got insulin binding to the alpha and beta. We get autophosphorylation. That's kind of step 2. We get recruitment of this insulin receptor substrate, or IRS docking proteins. And we can see that visually here. So I've kind of just I've hazed everything so you can kind of put it all in perspective, but we've phosphorylated here. We get this IRS docking protein. It phosphorylates, and therefore allows us to transmit things further down the line. The IRS protein contains multiple tyrosine phosphorylation sites that become phosphorylated after this insulin stimulation. So that phosphorylates the IRS1, results in, I'm just going to highlight this here, and I'll talk about it. So multiple tyrosine phosphorylation sites become phosphorylated after we get insulin binding. The IRS1 associates with IP3 kinase. So hopefully some of these terms are you're recognizing from last week or two. So we get PI3 kinase. So let everybody write a minute here. The PI3 kinase so here's our activated PI3 kinase. So if you want, and I would probably recommend this, so step 1, insulin binds to our alpha receptor, or our alpha subunits of our insulin receptor. The receptor autophosphorylates. That's step 2. Step 3, we get our IRS1 docking protein becomes phosphorylated. and that activates the PI3 kinase. So this activated PI3 kinase, we can see that we get some downstream activation of, activates this PIP3, and that then activates this AKT molecule. So downstream proteins include AKT, protein kinase C. So we get AKT phosphorylation. That does two things. This is kind of the next step where this AKT does part one. We get GLUT4 transporters inserted into the membrane. We'll talk about the importance of that, but we need these GLUT4 transporters in order to get these glucose molecules from outside to inside of the cell. So we've got to have these transported, and we'll look here, and I've got a couple pictures coming up here, but basically we've got these pools of GLUT4 transporters inside the cell, and they're not just in the membrane at all times. We've got them inside the cell, and we've got to tell them, these pools, to work their way from within the cytosol to the plasma membrane, become integrated. If we can increase the number of receptors, therefore we can then increase the amount of glucose transported into the cell. So this is the step in which insulin tells the cell to get GLUT4 transporters into the plasma membrane. It also then, what are we going to do with that glucose? We've brought it into the cell. What's next? So it also stimulates, we get glycogen synthesis activated. So we get two components. So through this binding of insulin to this receptor, we get this signaling pathway that leads to AKT that not only results in transporter relocation, but glycogen synthase activation. You should definitely be able to talk about those key steps in order. We have to stop right about here. As I've kind of pointed out in point 2, we really have a pretty limited understanding beyond that. We go to AKT, we protein kinase C, we get there, and really we're not sure what happens beyond that. But we know this is kind of the next major step. So if we talk about dysfunction within this pathway, and we'll talk about that at the very end of class, of where some of this can start to dysfunction. Well, we might have the insulin, we can't transport it in. You can start seeing how this pathway becomes critical in order for this response. We can give insulin, we can produce insulin, but we've got to make sure we have this effect, the actual outcome. That's one of the primary ways. We also know there's a MAP kinase pathway. So it runs in parallel to the insulin signaling cascade. It also is going to mediate things like cell growth and cell proliferation. So here's our insulin receptor, our alpha and our beta subunits. Here's our insulin molecule. We get autophosphorylation. Here's our IRS docking protein. We get through this pathway, here's our PI3K, here's our AKT. We talk about changes in glycogen synthesis, we got glucose uptake coming through this pathway that we just learned. Over here, this is the MAP kinase pathway. So insulin receptor, phosphorylation, RAF1, MEK, ERK1. So this is our MAP kinase down to our MAP kinase. All starts with this insulin receptor, but it has a different outcome, that being turning things on associated with cell proliferation and cell growth. So this one receptor, when it's bound to insulin, can result in multiple signaling outcomes. From a medical standpoint, if you're thinking about diabetes and those sorts of things, we're usually talking more about this cascade on the right, where the IRS, PI3K, AKT pathway. So this is what's happening at rest. We give insulin, we produce insulin. These are the responses that allow a cell to uptake that glucose and then use it. So what's happening during exercise? We know that exercise is a powerful therapeutic option for those who have difficulty controlling blood sugar. We know that it can really alter glucose uptake. So what do we know about it? So during exercise in the post-absorptive state, The contribution of blood glucose to ATP resynthesis is initially relatively minor, but as we start talking about the continuation of exercise and depletion, things go up from there. And we also know that it's exercise intensity dependent. So this is leg glucose uptake as a function of time at 65, 130, and 200 watts. And we can see two things here. One, that as the duration of exercise gets longer, we're talking 10 to 40 minutes in duration, we see a progressive increase in glucose uptake. So this kind of goes along this point here. As exercise continues and glycogen stores within the skeletal muscle is depleted, the contribution of blood glucose becomes more substantial. We can see that. In all conditions, it continually rises over time. But you can also see the most influential is the exercise intensity. We start seeing upwards of 3 to 4-fold increases in blood glucose during the higher intensity or higher work rates compared to these lower work rates, and definitely compared to our resting condition, which would be this first data point here. So what's allowing this to take place? What do we need to happen for this to occur during exercise? So we need to review just briefly, what are the 3 sites of regulation for skeletal muscle glucose uptake? Well, the first is glucose delivery. So this is going to be our supply. So perfusion, blood glucose concentration, those things, that's our delivery. We have poor delivery. If we have low blood glucose concentration, that's going to limit. We can also have to deal with the transport, so the membrane transfer. So this would be surface membrane permeability. So that's this transport in this. So we've got to have the GLUT4 transporters. We increase the density of those within a cell membrane, we're going to increase the transport. And then we have to have flux through the intracellular metabolism. So we've got to have basically our metabolism side. So these are the 3 potential sites of limitation for glucose uptake during exercise. Now, this is important because if you want to start talking about, where is the limiting step and trying to target that limiting step so that you can improve things, we need to look at some of the research. And just to boil it down, most studies suggest that glucose transport, so this point right here, so with the surface membrane GLUT4 transporter abundance, that is the rate limiting for glucose uptake in the muscle during exercise. We know that skeletal muscle, has multiple isoforms of glucose transporters, but during exercise, so if we're using exercise to say a therapeutic intervention here, the most important is GLUT4. And we know this because if you take an animal model and you knock out, go in and prevent that GLUT4 transporter from being expressed within these animal models. If you knock it out, it abolishes the contraction-stimulated glucose uptake, at least when we're studying in an animal model. Can't do that in humans, so it gets a little more challenging. So what's causing this GLUT4 transporter? If that's the limiting step, and we know that's important during exercise, what's turning it on? What's allowing the intracellular pools of these GLUT4 transporters to make their way to the plasma membrane so that we can actually see this glucose uptake? So this is whether you're dealing with it as a therapeutic intervention or if you're talking about in response to long duration exercise where you now need to rely on those blood glucose stores. So molecular events involved in GLUT4 movement within muscles, not only are they complex and based off these vesicle pools, the molecular signals that trigger increased glucose transport and therefore surface membrane GLUT4 are different when comparing the insulin and contraction stimulation. So what we've learned thus far, the response to insulin, that pathway is not how skeletal muscle increases GLUT4 transporters during exercise. So Currently, it is generally accepted that the underlying signals involved in contraction-stimulated glucose transport occur independently of the humoral factors that arise independently of humoral factors, and it arises from local factors within the skeletal muscle. So there's nothing in the blood, that the hormonal response to exercise does not appear to be the primary thing driving glucose uptake in response to muscle contraction. It's something within the muscle. So that means there's some signaling occurring within the skeletal muscle cell that's telling these pools of GLUT4 transporters to work their way to the plasma membrane. The first is calcium. We've already kind of briefly mentioned in the past, couple lectures that calcium is this great signaling molecule. We've got gradients within different cells, particularly skeletal muscle. That gradient allows it to change the intracellular concentration very quickly, so it makes a nice on and off switch. So during skeletal muscle contraction, we get these transient spikes in intracellular calcium. They occur with muscle contraction. We've known this for 30 years, and during that time, it's been hypothesized to be involved in this glucose transport. And it makes some sense, since intracellular calcium levels is related to the activity of the motor neurons. Signaling directly influenced by calcium can be considered a feed-forward regulation. You tell a skeletal muscle to contract, calcium spikes occur. That would then signal the cell as any feed-forward regulation that we're going to start needing glucose soon. Let's start working on these pathways. So calcium released from the sarcoplasmic reticulum interacts with calmodulin, gives us the calcium calmodulin-dependent protein kinase. We talked about that in one of the previous lectures. That's why I brought it up there, because it's critical within exercise responses to glucose movement, and that results in GLUT4 translocation to the sarcolemma. So let's look at this just visually. The text is nice, but I like the visual just a little bit better. All right, so here's our sarcolemma. We get our action potential, our exercise contraction, comes down our T-tubule, results in sarcoplasmic reticulum release of calcium, and we've learned in muscle phys and 335 and human body and so on and so forth, as this calcium goes down to result in skeletal muscle contraction. But it does more than just that. The thought process is that calcium binds to calmodulin, gives us our calcium calmodulin complex, and here is our GLUT4 pool. Results in translocation to the sarcolemma. Therefore, we've increased the GLUT4 transporter concentration within the sarcolemma, thus removing or improving this rate-limiting step in the movement of glucose across the plasma membrane. So that's that step, and you should know those steps. Okay, so that's skeletal muscle, and just in response to exercise. But we've already identified that there's an exercise intensity and duration dependent component to this. So we need to have a signaling pathway that not only responds to just muscle contraction on its own, but also responds to the energy state or the metabolism within that cell. So that results in our statement that energy sensing signaling systems is likely to also be involved with glucose transport with contraction. Zoom back in here for a second. And that's where AMP activated protein kinase, or AMPK, comes into play. We'll see this again come up when we start talking about the adaptations to different types of exercise. So what causes AMPK to become active, and what regulates its activity? Well, it can increase in response to the increases in the AMP, so adenosine monophosphate, to ATP ratio. It's going to respond to a decrease in the PCR creatine ratio. And in response to just 4 phosphorylation of AMPK kinases. Okay, so all these things we'd expect to be exercise intensity dependent. We also know that its activity is higher in skeletal muscle during exercise contraction, and its magnitude of activation is dependent on exercise intensity. So we've got this great signaling molecule. It's almost to think of it as an integrator that's going to respond not only to skeletal muscle contraction, but the intensity of the exercise, the metabolic state of the tissue is all going to be sensed based off this AMPK. It's not an on-off switch. It's very much like a dimmer switch. So it allows us to regulate signaling based off of all of these different factors. So a lot of it is from correlative studies. You know, it's a little bit hard to take humans and knock stuff out. The IRB gets a little fussy over those things. So you have to start looking at correlations. If AMPK is high, is GLUT4 transport or translocation also high? So we've seen that in individuals who have a high AMPK activity in response to muscle contraction or exercise, that they also have GLUT4 translocation and glucose transport within skeletal muscle. So a lot of this within human studies is going to be due to biopsies. You exercise, we take a small tissue sample, we rapid freeze it, and then we can go in and look at the different components. So what's happening? So go back to our image. Here's our calcium pathway. We know that's playing a role here. We know that we've got ATP required to drive this muscle contraction. So we get eventually AMP. We get from there AMP kinase. So this ratio and all of this is going to be dependent on the exercise intensity. Things like PCR, ATP, AMP ratios, so on and so forth, are all going to drive the signaling pathway through AMPK, through our GLUT4 transporter, and then again, improving glucose uptake. So I can't really fill in the gaps here from AMPK to GLUT4 because we just don't know. But we know that there's this calcium signaling pathway and this energy sensing pathway, all resulting in GLUT4 translocation. Since we're here, I'm also going to just make a small note about how AMP activity also has been correlated with increased fatty acid oxidation in skeletal muscle. I'm not going to highlight point A because this just kind of gets into the details. It just gets into the various details of this actual pathway. This lecture, I really want to focus on skeletal muscle glucose, but just know point 10 that it's also correlated with fatty acid oxidation. So it's likely playing a role here. We've got AMPK, which would, again, exercise intensity dependent, going to be sensed and regulated by a variety of factors. We get this signaling pathway where it's going to improve beta oxidation. So it goes beyond just the movement of glucose into the cell. There's some real integration going on here. We know that insulin nitric oxide signaling occurs both in normal physiology, but we start to see this come up in pathology as well. So in addition to the classical actions of insulin in coordinating glucose homeostasis, there's also a complete biochemical signaling pathway linking insulin receptor activation to that of endothelial nitric oxide synthase in the vascular endothelium. So it's playing two roles here. So this makes a nice true and false type question. So if you've not had muscle phys or 335, if you don't remember, ENOS, endothelial nitric oxide synthase, that's what's going to be required to get us from L-arginine to nitric oxide. That's going to result in not only vasodilation, we talk about that in response to exercise, but nitric oxide has a profound impact on endothelial health. It's an anti-atherogenic. You have poor NO bioavailability, you start talking about putting someone at risk of coronary artery and peripheral artery disease. Pretty substantial. So it's not just a vasodilator, it's also an anti-atherogenic component. So there's some real importance here when you start talking about insulin resistance, not only in its ability to uptake glucose, but also in some of the activation of things like eNOS and endothelial function. Okay, so I know this is a little wordy here, but we'll just go right through it. So insulin-mediated nitric oxide production requires activation of insulin receptor, tyrosine kinase. We've just looked at that before. Phosphorylates our insulin receptor substrate 1, so IRS1, that's our docking protein. So I don't have a figure because we've seen that. Leads to activation of PI3K. We then get AKT is then phosphorylated and activated, which then causes eNOS phosphorylation and subsequent nitric oxide production. Oh, I do have a figure. My bad. So insulin receptor. Here's our alpha subunits, our beta subunits. Here's our insulin molecule. IRS, just docking protein, PI3K, AKT. We've just talked about how AKT would then result in the GLUT4 translocation. AKT then also stimulates eNOS and improves nitric oxide production. Nitric oxide is going to have anti-apoptosis. We know it best as a vasodilator. It's going to also have some anti-inflammatory properties. It's going to work its way as being as an antioxidative stress. All pretty profound things when you start talking about cardiovascular health. If you start thinking about some of the issues when you start talking about insulin insensitivity, cardiovascular health, they're pretty linked. And not only are they linked in some of just the lifestyle and things going on there, there's actually signaling pathway associated with what's going on here. Insulin signaling and nitric oxide production, and all of these things are going to drive cardiovascular health. So, classically defined insulin resistance, pathological condition where insulin-stimulated glucose uptake and disposal is decreased. So, we basically, insulin-stimulated glucose uptake is decreased. We start talking about pathway-specific impairments in this PI3K-dependent insulin signaling. We start seeing reciprocal relationships between insulin resistance and endothelial dysfunction. So here's our insulin receptor, alpha and beta subunits, our IRS docking. Here's our PI3 kinase. We talk about it going down through AKT. Decreased eNOS, decreased insulin, that's going to promote insulin resistance, and endothelial dysfunction, all through the same molecular pathway. So if you can target this pathway pharmacologically, you can try and improve insulin resistance. Through this pathway, you're also going to improve endothelial dysfunction. And as I kind of already mentioned, endothelial dysfunction tends to be the first step in most all cardiovascular diseases. It's going to contribute to hypertension, or coronary artery disease. Pretty much everything starts with endothelial dysfunction and just snowballs from there. So when you start talking about insulin resistance, cardiovascular health, we see those comorbidities. They are linked through the same molecular pathway. Now, this is where exercise gets interesting, is because exercise, we've now learned, doesn't work through this pathway. We can contract skeletal muscle, and we can increase factors that are going to drive blood glucose down, because we're going to get translocation of our GLUT4 transporters through a different pathway than this. So if this is dysfunctional, we can still regulate blood glucose through muscle contraction, and exercise through various other ways, we'll talk about it later on in the semester, then improves endothelial dysfunction. So while they're dysfunctional through this pathway, we can then target it, either here or in different mechanisms. Questions about muscle glucose uptake? It's about as far much depth as we can get into because there's a lot of the rest of it's all a little bit speculative at this point. These are kind of the hard information. Okay, so